Hello everybody, this is Mr. Schultz, and this is the answer key for Module 9 quiz review on cellular reproduction and sexual reproduction. So, the first question on our review says, in the space below, fill in the parts of the cell cycle, then fill in the major events for each part of the cell cycle in the boxes below. And so, our cell cycle looks a little something like this. Okay, this is more complicated than the one on the notes. All it is it's a reorganization of the cell cycle and mitosis into one big diagram. Okay, and so the first part of the cell cycle that we're going to label is gap 1. Okay, and so here's gap 1, and then gap 2, or then is synthesis phase, so the S phase, and then gap 2, and all three of these, gap 1, the synthesis phase, and gap 2, G1, S phase, and G2, are all part of a process called interphase, okay? Which is just the cell cycle, the normal part of the cell cycle where the cell is doing normal cell stuff, growing, doing its job, things like that. Now, the last of it, down here, this is prophase, then metaphase, then anaphase, telophase. These are the 4 steps of mitosis. And then finally cytokinesis. And so this label should be mitosis right here, and then PMAT, and then cytokinesis. And so that brings us down to the major events. And so the first major event that we have is going to be the G1 phase, okay, gap 1. And so the gap 1 phase is going to be that cells grow and does normal cell function. So the cell is just doing its normal life, okay? It's just, you know, if it's a heart cell, it's doing heart cell stuff. If it's a lung cell, it's doing lung cell stuff. And then, for the next step, we have the S phase. Now, during the S phase, the cell makes a copy of its DNA. The S stands for synthesis. It means to make something, okay? So in this case, it's making DNA. The next one is G2, which is gap 2. During gap 2, the cell keeps growing to get bigger and keep doing its job, but it's also getting ready for mitosis. It's getting ready to divide. And that leaves us with mitosis. Mitosis is when the chromosomes divide. So we oftentimes think of mitosis as the cell splitting, but technically mitosis is actually the chromosome splitting, and then cytokinesis is everything else splitting. Okay, so the cell divides its organelles and cytoplasm, and the cell splits into 2 new cells. And so that's question number 1, which is looking at the cell cycle and what happens in each part. For question 2, it says, during our meiosis notes, we talked about diploid and haploid cells. Explain what diploid and haploid mean, then explain in which types of cells you would expect to find these types of cells. Which is kind of a weird way to ask it. But basically, what kind of cells do you expect to be diploid and haploid? So if we look, our first one is going to be diploid, okay? And so in a diploid cell, we have cells that have 2 of each pair of chromosomes. 2N is the way that we wrote it in our notes. So 2N is diploid. I like to think of diploid as being normal, okay? Everybody's normally got 2 of every chromosome. And so the example are just normal body cells. The vocab term that we use is somatic cells, okay? And then we have the haploid cells, okay? In a haploid cells, they only have 1 of each chromosome. We label them as an N instead of 2N, and then have half the normal number of chromosomes, haploid, half. And so what kind of cells are those? Those are the sex cells and the gametes. So for question 3, it says, hey, looking at your notes, identify during which phase of meiosis does each event occur. And so for the first one, it says cytokinesis divides the cells. Well, it turns out in cytokinesis, this happens during both meiosis 1 and meiosis 2, which kind of should make sense because during meiosis 1, you're having double the chromosome split down to the normal number, and then meiosis 2 is taking that normal number of chromosomes and splitting them in half, so each cell now has half. Crossing over is when genes swap. It happens during meiosis 1. Specifically, it happens during prophase 1 of meiosis 1. Homologous chromosomes, they separate during meiosis 1. The daughter cells are diploid, so we have daughter cells twice. Diploid, normal numbers, is after meiosis 1. The daughter cells are haploid. Well, that happens after meiosis II. And then finally, there's a typo in our quiz review. It's supposed to say sister chromatids, not homologous pairs. Homologous pairs is not correct. Sister chromatids is what we're talking about. It's where the identical copies of each chromosome get split, and that happens during meiosis II. Okay? So that's that. That's just identifying. Odds are, if we ask a question like this on the quiz, it'll probably be a diagram with parts labeled, and you just have to look for it and read the labels correctly. For 4, it says, hey, how are the cell cycle and cancer related to each other? In yours, there's space. On my key, there isn't, unfortunately. But cancer is what happens when the cell cycle goes out of control. So on the first page of our review, we had the cell cycle picture. What happens is there's these checkpoints, and if the checkpoints are ignored because of a mutation or something, the cell just keeps going through the cell cycle without ever stopping and making sure that it needs to keep going, because most cells don't need to divide over and over again. And so what happens is it just keeps going through the cell cycle over and over again, and it starts to grow out of control, and now we end up with cancer. Now, 5 says compare and contrast mitosis and meiosis. And so in mitosis, we have 1 division that occurs, and in meiosis, it's 2 divisions that occur. In mitosis, DNA replication happens during interphase. In meiosis, DNA replication happens before it starts. It actually happens during interphase. It's just the book, for some reason, doesn't want you to get confused. It makes you want to... It's like it wants you to think that there's some other thing going on instead of an interphase before meiosis, but it's still technically interphase. In mitosis, you end up with 2 identical daughter cells that are formed, and in meiosis, you end up with 4 unique haploid cells that are formed. And then during mitosis, your daughter cells are genetically identical, and in your meiosis, daughter cells are genetically unique. In mitosis, crossing over does not happen. In meiosis, crossing over definitely happens. It's one of the things that creates uniqueness in the cells that we were talking about before, the daughter cells. And so during mitosis, we also have body cells only, and in meiosis, we're making reproductive cells only. We call those gametes or sex cells. And then finally, it says what happens, or what's the main function of mitosis? It's for growth and repair. For meiosis, it's for reproduction or production of gametes, so making those sex cells, those sperm and egg, if you will. Okay? So that's question number 5, comparing mitosis and meiosis. For question 6, there's the word bank for question 4. Question 6 is a compare and contrast chromatin and chromosomes. The review that I'm using is a little bit old. I updated it since last year, so it's going to look a little different. But for my chromatin, they're loosely packed. They're formed for interphase. So interphase is when we see chromatin. And they're hard to move or separate because they're kind of like a big pile. Okay, they're loose. Here's my drawing of one. And then for chromosomes, they're tightly packed. They form before the cell splits, so during prophase 1, and they easily move. They're easy to move or separate. So basically, they can be divided. And here's my little doodle of one. That's a chromosome. During our mitosis pictures, we also saw them sometimes drawn like this. That's a chromosome as well. Okay? Now, what do they have in common? Well, they're both made up of DNA. And they're both wrapped around histones. So that's another example where, oh yeah, they both are wrapped around these proteins that kind of help hold them together and keep their structure normal. All right, for question number 7, how do living things use mitosis, meiosis, and fertilization to maintain their species? By the way, this is a good one to study. Okay? Hint, hint. Now, fertilization causes the formation of a new organism, a fertilized egg. The egg will undergo a bunch of mitosis to grow and develop. We call eventually it'll turn into an organism, which is called like differentiation when stem cells become normal cells. We'll talk about that in the next question. Once the organism becomes an adult, it will start to produce gametes. Through meiosis, this organism can now also reproduce, which allows the species to continue. In other words, it can now go through meiosis itself, where it's going to make sex cells, which is going to then combine with other sex cells to make a new organism, which then allows that organism to grow and develop and become an adult and make its own gametes, and so on and so forth. Okay, there was a diagram in our notes that looks like this. Okay, in other words, this answer got pulled from here, and you should definitely study that a little bit. Okay, now question number 8 says, what is differentiation? How does it relate to what is happening in question 7? Well, differentiation is when stem cells are given a signal to develop into a more specialized cell. While the baby is growing, it goes through mitosis, a lot of mitosis. These new cells will be given a specific role through differentiation. In other words, they're given a job. The new cells will have a specific structure and function. In other words, a heart cell needs to do heart cell stuff, like help pump blood, and it needs to have a very specific shape, and it needs to be able to do its job, as opposed to a lung cell that'll have a different function. In this case, it probably is pulling oxygen out of the air and putting CO2 back into the air, and its structure needs to look different, so it needs to be a different shape. And so that's really important to understand. Differentiation is when a stem cell that has no job or major role gets turned into an important specific cell. Okay, so differentiation is where that baby in question number 7 is growing and developing. Well, that's what differentiation is it's changing the cell into something that's useful and unique. Now, for question number 9, question 9 asks you to calculate the surface area to volume ratio for each of the following cells, then answer the follow-up questions. Now, this question seems really intimidating at first, and part of the reason is because I'm asking you to do some math in the review. On the quiz, there is no math asked of you. All you need to do is compare the numbers and read the diagram and the data table to figure out the answers. But for the quiz review, I like to make it a little more challenging so that we have a little bit more to do. And so our side length for our box A is 2 centimeters. To figure out the surface area, it's 2 times 2 times 6, so we end up with 24 centimeters squared. Our volume is 8 centimeters cubed, and our surface area to volume is 24 to 8, which simplifies down to 3 to 1. And you have to remember, surface area to volume, the bigger the first number is compared to the second number, the bigger the surface area to the volume is, the more efficient the cell is, and the happier the cell is. And then we go to box B. Our cell B is 3 by 3 by 6, so it's 54 centimeters squared. And then its volume is 3 times 3 times 3, length times width times height. And we end up with 27 centimeters cubed. And that gives us 54 to 27, but that's not simplified. And so what I'm going to do is I'm going to simplify that down to 2 to 1. And then we have the 4 by 4 by 4 centimeter cubes. So this is C. And so it's 4 times 4 times 6 for the surface area, because it's length times width times the 6 sides. And that gives us 96 centimeters squared. And then we do 4 times 4 times 4, which gives us 64 centimeters cubed. Our first initial number is 96 to 64. If we simplify that by removing a 16 from both sides, we end up with a 3 to 2. Okay, and I think actually you can take a 32 out of there, actually. So I divided both sides by 32, and I end up with 3 to 2. So if you notice, this was 3 to 1, this is 2 to 1, and this is 3 to 2. The difference between this cell and this cell might look like just 2 and 1, but this cell is more efficient than this one, which is more efficient than this one. And then finally, cube D is 5 times 5 times 6. We get 150 centimeters squared. 5 times 5 times 5, because length times width times height gives us 125 centimeters cubed. And then finally, 150 to 125. I end up with a 6 to 5 relationship. And there's actually a trick to this, by the way. When you do the math and you write everything out, you'll notice that 5 times 5 times 6, 5 times 5 times 5, if you ignore these first two 5 by 5s and 5 by 5s, you get a 6 to 5 relationship. Over here, you get a 6 to 4 if you ignore the 4 by 4s. but that reduces down to 3 to 2. Same thing over here. 6 to 3 is a 2 to 1 relationship, and over here, this is a 24 to 8. You can divide both of those. That one you can ignore the 2's, and you get a 6 by 2, which is a 3 to 1, which is pretty cool and a nice way to kind of figure it out. So, for question number, for letter A, I don't know why I'm covering this up. Letter A says contrast the surface area to volume ratio of cell A and B. How are they different? Well, A has a greater surface area to volume ratio, which means it can do diffusion better. In other words, 3 to 1 is better than 2 to 1, which is better than 3 to 2, which is better than 6 to 5. This cell would really struggle, by the way. Which cell is probably doing its best to survive? Cell A. How do we know? Because the surface area to volume ratio is better. And then see which cell is probably doing the worst? Well, that would definitely be cell D, right? 6 to 5. And instead of doing apoptosis because it's like struggling and it might die, instead what it might do is it might just do mitosis. It's going to split, and it's going to cut its surface area back down to a more manageable size. That's all mitosis is basically about. When it comes to cells getting too big and they get efficient, the cell will split, and now they're more efficient. Okay? A lot of cells don't even split. They just grow to a certain size and stop. We talked about that earlier in the year. It's called G0. All right, for 10, sometimes during meiosis, mistakes are made. It is important to understand what happens to cause these mistakes. In the diagrams below are models of meiosis in dogs. First, let's look at normal meiosis. During the first division of meiosis, homologous chromosomes are separated. During the second division, sister chromatids separate. Draw the number of chromosomes in the resulting cells after meiosis 1 and meiosis 2. These, by the way, these little shapes, they're supposed to look like this. Okay, I'm just not very good at Google Draw and struggle to make a proper cell that looks appropriate. So it should really look like this inside the cell, and that's an X and an X. Okay, we're just looking at the sex chromosomes for now. Dogs, by the way, they have quite a few chromosomes. So this is just one of the chromosomes. So during meiosis 1, the homologous chromosomes, the two X chromosome pairs, because these are technically 4 X's here, because a normal single X looks like that. And so what this really is it's just 4 X chromosomes. They're just combined together. They're stuck together for now, okay? And so what's going to happen during meiosis 1 is we're going to split these two X's apart. Basically, we're going to cut them down the middle, and one is going to go this way, and the other is going to go this way. And so it's really kind of important to know that this X is technically 2 X chromosomes that are identical to each other. We call them sister chromatids. Okay, so these, if I were to draw it much bigger, okay, this and this are called sister chromatids. These are called homologous chromosomes. Okay, it says homologous chromosomes here. And the reason they're homologous is this X is from mom and this X is from dad, and they aren't identical to each other. They code for the same things because they're the same chromosome in terms of like what they code for, what genes are on them. But dad's X chromosome and mom's are probably not going to be identical. And so what's happening during meiosis 1? The homologous chromosomes separate. They go their opposite sides. In meiosis 2, these two X chromosomes now need to split. And so this one's going to go here, this one's going to go here, and then over here, the same thing. Okay? So these are all, you can kind of think of them as like 2 X's here. This is technically 2 X's here. And then down here, each one, there's only 1 X. Because when they come together with a sperm, you want to only have 1 sex chromosome in each cell beforehand, each gamete. Otherwise, you're going to end up with extra chromosomes and things aren't going to go right. And that's what this part B is all about. Sometimes non-disjunction takes place. During non-disjunction, the homologous pairs did not separate. What happens to the cells at the end of meiosis 2 when non-disjunction occurs during meiosis 1, draw what will happen. So before, these two guys got separated and then they split over here. Okay, so they got divided up into two different cells. Over here, instead, the X and the Y, keep in mind there's technically two X's and two Y's here, they're going to get separated into this cell, and this poor cell doesn't get anything. Okay, so all the X's and the Y's end up over here. Okay, and so what's going to happen is you're going to end up with a splitting, and you're going to end up with an X and a Y in each. That wasn't a very good Y. But here's my X, here's my Y, here's my X, here's my Y. Oh, and hey, look, a whole lot of nothing in this one. Okay? So if this was a human, we would have 22, because the normal number of chromosomes in a human is 23 for a gamete. And this one's missing one, so it's going to be 22. And over here, it's got an extra one. That's going to be 24 instead of 23. So in human gametes equals 23 chromosomes. Okay, that's our N number, so N equals 23. In the case of this one, it's one short, so N equals 22. And over here, there's an extra, so N equals 24. And so if we were to say, instead of humans, let's look at a dog, a puppy. So suppose the female from part A has puppies with a male from part B. You're like, wait a minute, we were talking about humans before. Well, we're talking about dogs now. Okay? How many chromosomes would each puppy have from part B? So a normal dog has 39 chromosomes, including the two sex chromosomes. So that's supposed to be, I believe, 39 chromosomes in their gametes. But hold on one second. I'm going to double check. I had to do some quick research. 39 is the number in the gametes. The normal number is 78. Okay, that's probably where the 39 came from. So a normal dog has 78 chromosomes. Compared to us, we only got 46. So what would happen if you had a sperm from this dog and a egg from this dog? How many chromosomes would it have? Well, if this gamete's supposed to have 39, but it's got an extra one, it's 40. So normal is 39 plus 39, okay, which equals 78 total. So that's N, N, and 2N. Now, if it took one of these sperm cells where it's got an extra one, now non-disjunction. So in non-disjunction, we have 24. Sorry, not 24. It's an extra one, so it's 40 plus 39 equals 79 total. So that's this one. Okay? And then if we were to take this one, non-disjunction, this one's short one. So instead of having 39, it's got 38. But it gets a normal egg from mom, so it's going to be 39 for the other. And so now it's got 77 total for its 2N. You can't see that. So non-disjunction with this sperm is 40 plus 39 for 79 total. And non-disjunction for this sperm, we end up with 77 total. And so what's going to happen is this puppy is either going to be one chromosome short or one chromosome extra. And this is going to cause some sort of issues along the lines of probably developmental issues with the puppy before it's even born. And sometimes puppies have this happen. Cats do too. where they have puppies that have the wrong number of chromosomes, and a lot of times they don't get born. They get stillbirth. So, and that happens sometimes with puppies as well as kittens and people, believe it or not. So this is our quiz review. If you have any questions, don't be afraid to ask them before our quiz tomorrow, and please make sure you spend some time studying, and good luck.